In this video, I'll present information from Chapter 11 of Human Anatomy and Physiology, 11th edition. This chapter focuses on nervous tissues. In particular, this video will cover synapses and an introduction to those synapses. So what is a synapse? A synapse is a junction between a neuron and a second cell. This is the location of information transfer between those cells. You can see here that the neuron is always the presynaptic neuron, meaning it comes before the synapse. And the synapse is this junction between the two cells. So we have a presynaptic cell before the synapse, as well as a postsynaptic cell. There are always those two cells involved in a synapse. And in a chemical synapse, they are separated by a space called the synaptic cleft. There are actually two types of synapse, although a chemical synapse is by far the more common of the two, and so that's the one that we'll focus on in this course. However, there are also electrical synapses. They're quite rare, and instead of being separated by those synaptic clefts, the presynaptic and postsynaptic cells are actually directly connected by these structures known as gap junctions, which are basically pores that form between the two cells. So the advantage of having these gap junctions is that this allows for extremely rapid communication. And that's because the action potentials can cross directly through those pores, through the gap junctions, from the presynaptic cell to the postsynaptic cell. This also means that there doesn't need to be any translation between an electrical signal and a chemical signal. The signals stay electrical throughout the process, from the presynaptic to the postsynaptic cell. So again, this allows for very rapid communication. The chemical synapses are by far the most common. And in these, again, the presynaptic and postsynaptic cells are separated by a synaptic cleft. These chemical synapses use chemicals called neurotransmitters that come from the presynaptic cell to stimulate the postsynaptic cell. The big advantage here is that you can have more complex messages. So instead of just having one signal coming from the presynaptic cell, you may have multiple signals, or you may have multiple presynaptic cells sending signals to the same postsynaptic cell. One thing just to compare with an electrical synapse, so electrical synapses, again, they remain electrical throughout the process. Chemical synapses, you have electrical signals running through the presynaptic cell. Those are action potentials. They then turn into these chemical signals that cross the synaptic cleft, and then those chemicals cause further electrical signals to occur in the postsynaptic cell. So now let's focus on those chemical synapses, and that will be throughout the rest of our discussion of this chapter. So synaptic communication at chemical synapses can be broken down into multiple steps. We've already hopefully looked at a video on action potential propagation down the axon of a presynaptic neuron. And now we get to the synapse. So we're talking about synaptic communication, it's chemical synapses. So step one, that action potential propagates down the axon, and it actually arrives at the axon terminal. That action potential is then going to stimulate voltage-gated calcium channels to open. And calcium is now going to flow into the synaptic terminal of this presynaptic neuron. That calcium influx will trigger the release of neurotransmitters into the synaptic cleft. So this is the presynaptic portion of signaling at a chemical synapse. So now we can go into that synaptic cleft. So the neurotransmitter has been released from the presynaptic neuron, and it now diffuses across the synaptic cleft. Once it diffuses across, it reaches receptors on the postsynaptic cell. So here you can see receptors, and they bind to that neurotransmitter. And those receptors, they're called ligand-gated ion channels. And now those ligand-gated ion channels open in response to that neurotransmitter, and that allows for ions to flow in and or out of the cell. And depending on the ions that flow and in which direction they're flowing, this can cause the postsynaptic cell to either hyperpolarize or to depolarize. And this will be a graded potential. So you can have a hyperpolarizing or depolarizing graded potential in the postsynaptic cell. And typically, those postsynaptic cells are other neurons or maybe muscle cells. So now we've looked at the synaptic cleft and postsynaptic response to communication at chemical synapses. So the final step of synaptic communication at a chemical synapse would be the termination of neurotransmitter effects. This is vital because the nervous system is meant to be a rapid and short-lived communication system. This allows it to be very specific in its effects. And so we need to quickly terminate neurotransmitter effects. Remember, there's all this neurotransmitter binding to postsynaptic receptors, causing hyperpolarization or depolarization in the postsynaptic cell. We need to get rid of that quickly to terminate the effects. And there are three basic methods for doing that. We have first reuptake. This means that once that neurotransmitter is released into the synaptic cleft, it then gets quickly returned, either returned to the presynaptic cell, so it moves back into the presynaptic cell, or it moves into the astrocytes that are also surrounding this synaptic cleft. So that's reuptake. The neurotransmitter can also be terminated by degrading it. So this is a breakdown caused by enzymes. If you remember back to learning about skeletal muscle fiber stimulation by nerves, hopefully you still remember that acetylcholine was degraded by an enzyme called acetylcholinesterase. And so this allows for that acetylcholine to have very rapid and short-lived effects, so that when you ask for your muscle to be contracted, it contracts but doesn't get stuck in the contracted position. And then finally, we have a third method of termination, and that's diffusion. This is probably the simplest because this just involves the neurotransmitter diffusing out of the synaptic cleft to the point where there just isn't enough of it to activate the postsynaptic cell. So those are the three methods of terminating neurotransmitter effects. And now I'll just leave you with a question.